In part 3 of our videos on the evolution of life, we're going to take a look at the eukaryotic cells now. And at this point, this is when our prokaryotic cells are going to start to evolve into the more complex eukaryotic cells. And remember, we're talking about a period of probably 2 billion years here of this lifespan for the planet before we actually hit this part where we see these eukaryotic cells appearing. And when we look at this process, we're going to start focusing on the different kingdoms of eukaryotic cells that are out there. And we'll start out with the Protists and work our way up to the fungi here. And if we look at this process, well, how could these eukaryotic cells evolve? Well, we can look at the origins of that. They came from prokaryotic cells. So what are the major differences between prokaryotic and eukaryotic cells? We know about their chromosomes are circular versus linear in eukaryotic cells, but the other major difference is these membrane-bound organelles. So how could you get these membrane-bound organelles to form? And one hypothesis, because again, we cannot prove that this happened, but it is an educated guess, is that you have your ancestral prokaryotic cell that was out there with its DNA floating around in the cytoplasm. And then you started to get invaginations of that membrane or folds that appeared that started to protect that DNA chromosome inside of the cell. And eventually these membrane folds started to go back and forth, and that is a possibility for how we could get this endomembrane system that is surrounding the nucleus. And then remember, things like the endoplasmic reticulum with the smooth and the rough forms, those are actually continuous with the nuclear envelope there. So those would just be some extra folds that developed in the nuclear envelope that allowed the cell to start doing more and more complex processes. Remember, when we think about this idea of evolution, we have to think about the process of remodeling existing structures that were already in the cells, and this process is not happening overnight. Cells do not evolve what they want. They evolve through random mutations that occur that produce some sort of change in those cells that allows them to be favored in nature. That process of natural selection is what drives evolution that is out there. So this is one possibility for how we can get this nuclear envelope and then this endomembrane system within the cell, within the eukaryotic cells that are out there. But what about the mitochondria and the chloroplast? And this is where we see a really interesting idea that was proposed by a female scientist in basically the 1960s, and her name was Lynn Margulis. And what she proposed is that by looking at some similarities between the prokaryotic cells that were out there and mitochondria and chloroplast, she came up with this hypothesis that these particular organelles actually came about through a process known as endosymbiosis. And when you look at endosymbiosis, you're looking at endo is inside, and symbiosis is life that two organisms that can live together. And the hypothesis was that somewhere in the past that there was a prokaryotic cell that could do electron transport chain and was able to grow aerobically, and this cell was engulfed by a larger prokaryotic cell that had evolved that process of the endomembrane system. And now you get this cell that was living inside of the other cell. And if you think about this process, it's being eaten, but it's not being digested inside of that cell. And it turns out that these cells benefited from this process. The one that was inside got protection, and the bigger cell here was able to evolve, and it started to get more and more energy from that, what was now the mitochondria, and these two cells living together were able to benefit from this process. And when we look at our photosynthetic cells that are out there, the hypothesis is that these guys developed because a photosynthetic prokaryotic cell was also engulfed inside of these cells. Because when we look at our photoautotrophs today, we find that they not only have chloroplasts, but they also have mitochondria that are out there. And so that could give us our photosynthetic eukaryotic cell. And when we look out there today, we can actually find populations of amoeba-like cells in the ocean that are actually going through this process. And when you look at the DNA inside of their mitochondria, it resembles the DNA in the surrounding area. So this process of endosymbiosis might still be occurring today. And when you look at the evidence for this hypothesis, it turns out there are some very distinct similarities between prokaryotic cells and the mitochondria and chloroplasts. You've got similarities in terms of their size in that 1 to 10 micrometer range, and also their shapes. We find inside of our mitochondria and chloroplasts that they have circular chromosomes like prokaryotic cells, and there is homologous DNA sequences in prokaryotic cells and the mitochondria and the chloroplast. So again, that evidence for evolution that is out there based on similarities or homologies at the DNA level. When you look at how these things divide, they both divide by binary fission, and the final piece that we can see is that double membrane that we see around our mitochondria or our chloroplasts. Well, the inner membrane actually has folds that resemble the outer surface of bacteria, and within those folds are the energy mechanisms, the electron transport chain and the ATP synthase that are there. And there is a certain amount of homology between these at the DNA and at the protein level that is out there. So now we've got our possibility for how these eukaryotic cells could have started to evolve. And if we look at our phylogeny or this evolution of these different types of life that is out there, you can see that it is pretty complex. We have a common eukaryotic ancestor over here, and that ancestor diverged to give us all these different branches on our tree of life. This is where we are down here as the animal kingdom, but you can see there's a lot of different groups, and these have been grouped together in what are called supergroups based on evolutionary relationships that we can track down by looking at the evidence that is out there. And when we look in this kingdom over here of Protists, it turns out there are two major groups or subkingdoms of life that are out there. They're both eukaryotic, but they are very distinct in terms of their differences. The protozoan cells there, proto is first, and then think first zoo. These are animal-like single cells that are going to move around in basically wet environments out there. They do have animal-like membranes around the outside, and they are heterotrophs, so they're going to be able to get their carbon by basically absorbing nutrients by breaking down dead organisms or their waste products. The other major group that we see in our kingdom Protistas out there are the algae, and these are more plant-like. There are single and multiple cellular forms of them, but they are different from plants because they lack true tissues. When you look at the algae, they are photosynthetic, and many of them have cellulose for their cell walls, so very similar to plants. Some of them do have some differences. They have different chemicals that can make up their shells. And what's even more unusual when we look in this kingdom Protista is that some of these organisms are chemoheterotrophs at night and photoautotrophs during the daytime. So when you look in this kingdom of the Protists, this is really the junk drawer where biologists would throw all the organisms that didn't fit into the other kingdoms that are out there. And only now that we can look at the DNA level are we starting to unravel some of these relationships that give us all these different kinds of supergroups. And people are starting to propose that maybe these should be different kinds of kingdoms of life that is out there. So we're going to take a look at the Protists that are out there. And there's probably about 100,000 different living species of Protists that have been identified. And they're grouped together based on different lifestyles. So again, this is not phylogeny. This is basically just putting them into some categories for our own convenience. And again, just like with the bacteria, we want to consider their idea of reproduction because it is biology. And when you look at the different organisms that are here, they can basically alternate between asexual and sexual reproduction. If they are doing asexual reproduction, they will divide by binary fission, but again, it's that process of mitosis. Some of them can do multiple fissions where they basically break off a little part of themselves. Others can bud, where a small cell forms on the mother cell through the process of mitosis. And we can also, like I said, find sexual reproduction. So these cells are able to do meiosis at the single cell level, and they're going to produce haploid cells that can basically fuse together to make more diploid cells. So when you look in this kingdom, their reproduction is all over the place. One kind of similarity to the prokaryotic cells is when nutrients are scarce or the environment changes, they will often do sexual reproduction, but they can also shift from what's known as a trophozoite form, which is the actively reproducing form, and they will start to form what are known as cysts, which are kind of the equivalent of an endospore. It's a dormant cell with a protective coating around the outside. If we take a look through these protozoans, we can see they're all over the place in terms of shapes and sizes. And here we can see our flagellates. These are different groups of protozoan or animal-like cells that have flagella on the ends of their cells. And these include things like the diplomonads that actually have 2 nuclei, and then this euglenozoan over here that is actually able to swim through your bloodstream. And again, these are just going to be feeding off of organic molecules either in your blood or in your intestines. We've got our amoeba-like cells over here, and these guys basically move around like little blobs, and they extend parts of their cytoplasmic membrane, and that's known as a pseudopod or a fake foot. And over here you can see they've ingested some food via the process of phagocytosis, so basically engulfed it with their cell membrane, and they bring it in and they're going to fuse it with lysosomes and digest these little food particles inside of a vesicle inside of their cells. When you look at the amoeba that are out there, there are some free-living types like this one that's known as Amoeba proteus, and we can find other amoeba-like organisms that have a protective shell around the outside, and these are the little pseudopods that they are sticking to the outside that will be able to grab on to bacteria and other small cells and basically drag them in. So this is kind of like an amoeba that has an armor around the outside. Here we can see some other protozoans. The apicomplexans are parasitic cells that have this weird apical complex on the end that they actually use to drill and enter into other cells that are out there. So that is a characteristic of this group. And this is an example of the Plasmodium, which is the organism that actually causes malaria when it invades cells in your body here using that apical complex. And here are some really beautiful examples of some ciliates. And these organisms are characterized by the presence of little cilia on the outside of the cells that are basically like short flagella with multiple copies of them. And they'll use that to swim around and also to stir up the water and drive some food towards the cells that are out there. So really some interesting kinds of organisms here when we look at the protozoans. There is a related group in this kingdom Protista that are known as the amoebazoan slime molds. And they're really kind of strange. They are amoeba that actually resemble fungi during certain reproductive stages. And this resemblance is not because they have a common ancestor, but what's known as convergent evolution. They converged on a similar kind of solution as the cells evolved. And the slime molds are going to ingest their food using phagocytosis, and they lack cell walls. So they are indeed different from the fungi that are out there. And there's 2 representative examples of our slime molds. The cellular slime molds you can see in the amoeba stage, and these, when they run out of food, will actually start to aggregate, and they form this little slug-like colony thing that kind of crawls around, but this is like the size of a fingernail clipping. And inside of here, they're going to form this reproductive structure. And on the end of it, you will find their version of a spore that can get released and then float around in the environment until it finds a favorable environment where there's more water or food available. And this one is a plasmodial slime mold. And these guys are decomposers that are found on the floors of forests. And a plasmodium is basically a big mass of amoeba-like cells. And these things can get huge, and they're basically going to decompose things. So you will often find them on the floor of a forest, basically dining on trees and dead material that is out there.